right. Well, I'd like to welcome everybody to the May Dunlevie seminar series. It is my pleasure to introduce our speaker today, Mike Angelo.
Virginia, let me just make a few comments
yes.
about
So sorry.
No, absolutely. Just so that we give a couple of updates and then we will jump into this awesome presentation. Anna and Rebecca, I'm just going to go ahead and start with that as people come in. So just a few center updates. The Fetal Therapy Symposium was held on May the 2nd and went beautifully with really great attendance and a lot of wonderful presentations. And special thanks to the fetal therapy lead physician director, Dr. Yeri Blumenfeld, and our director of research, Anna Gearson, and our program operations specialist that keeps everything together, Rebecca Mobley, leading and organizing a really great program. Our spring 2024 seed grants, the applications are now closed. The review process is underway. Thank you to all the reviewers. We fund 2 awards, $35K each for 12 months, starting July 1st, 2024, and applicants should be notified by June the 12th. So our next speaker is going to be Dr. Michelle Williams, visiting epidemiology professor at Stanford University and the Joan and Julius Jacobson Professor of Epidemiology at the Harvard Chan School of Public Health. And she will be discussing perspectives on uncovering triggers of acute perinatal outcomes, an epidemiological and molecular approach. And we're all looking forward to this fascinating presentation. And I will hand things over to my colleague, Dr. Virginia Nguyen, to introduce today's speaker.
Wonderful. Great. So it is my pleasure to introduce Mike Angelo, associate professor here at Stanford in the Department of Pathology. Dr. Angelo received his MD PhD from Duke University with a PhD in electrical and computer engineering. Then he completed a residency at UCSF and did his postdoctoral training here at Stanford under the auspices of Gary Noland. And Mike actually really pioneered a new technology, being able to look at multiple analytes in pathology specimens using tagged heavy metals and an ion beam laser, which was really transformative for being able to look at tissues in a much more comprehensive way. He has interests in cancer biology and more in immunology, and more recently has gotten intrigued with the placenta and the maternal-fetal interface and a lot of the immunology and cancer traits that trophoblasts have at this interface. So it is with great interest I look forward to your talk, Mike. Welcome.
Thank you. Let me get my presentation up here. So full disclosure, Virginia and I work together, so that's why it's such a friendly introduction. So
it's well deserved.
Oh, well, thank you. So yeah, I'm really excited to talk to you guys today. And I find, like, no matter what audience I'm talking to, I probably end up like annoying somebody in the audience because I'm off topic for something that's, so this is, maternal fetal medicine, and still I've got cancer stuff in here, but it all holds together. And I'm going to kind of go into why I think that there's a lot of synergy to be gained from looking across fields to understand sort of commonalities and how immune tolerance is induced in these different settings. And so to start off, and these things kind of translate to things with the maternal-fetal interface as well. But it's sort of kind of what got us started looking at this is that, was with tumors, is that tumors don't really exist in a vacuum. They're really a part of this broader, more complex ecosystem that involves, dynamic interactions with neighboring immune cells, stromal cells, blood vessels. And it's really this interaction and sort of the emergent properties that come out of this ecosystem that sort of dictate what the composite function is. And the other part is that the tumor microenvironment, the structure really isn't random. And so, which is probably obvious to a lot of people, but it's but the good thing about this is that the location and function of different cells within this environment, it really almost serves kind of like as a historical record of what led to that state and what the current state is, and we can exploit those features to be able to get kind of a deeper understanding of what's going on, which can be very helpful when you want to look directly in sort of primary human material. So as we alluded to before, a lot of what we actually look at kind of three main areas in my lab. The first is obviously cancer in the context of breast cancer and brain tumors. The other is sort of understanding the crosstalk that the pathways that have been identified in that setting have in pregnancy and also in tuberculosis. And there's actually a striking amount of overlap in sort of how immune tolerance is induced across these three different areas. And so today I'm going to show first sort of the approach that we take to sort of use spatial information to inform what's going on in each one of these scenarios. And then I'm going to start by first showing sort of examples of how we've applied those in this context of a few brief examples of tumors to sort of illustrate why taking this approach with sort of primary human tissue and taking a deep dive on sort of how they're spatially organized can be fruitful. And then last, I'm going to just kind of show a study that we published recently looking at the first 20 weeks of pregnancy at the maternal-fetal interface, and hopefully that'll make the case of the striking parallels and sort of what's going on in tumors and what's going on in pregnancy. So to get at these questions, we really we look primarily at human material, and we use three techniques to get at questions of how they're organized. The first is one that we developed a few years ago that we use for spatial proteomics, where essentially, if you're familiar with like IHC, it's very, very much the same. The antibodies are tagged, instead of being tagged with fluorophores or chromogens, they're tagged with metals. And then it's read out, instead of with a light microscope, we image these tissues with an ion microscope. And so, and the benefit of doing it that way is that we can multiplex around 40 to 50 different targets simultaneously. And so that gives us sort of a targeted look at the protein expression and spatial distribution at a subcellular level. The other approach that we use is spatial transcriptomics. We've used mainly ROI-based techniques up to recently, and we're now starting to look at some of the newer technologies that actually allow you to visualize hundreds of transcripts at a subcellular level within individual cells. And then lastly, the other approach that we use is something called MALDI, or matrix-assisted laser desorption ionization. And essentially, the advantage with MALDI is that, unlike the other two approaches, which are targeted, so you kind of have to have probes, either oligonucleotide probes or antibodies, MALDI is a de novo mass spec technique, which means that more complex chemical characteristics of the tissue can be ascertained. And we're using this centrally right now to really understand a critical but often neglected post-translational modification of proteins, which is glycosylation. And so I'll talk more about that in a little bit. So irrespective of the technique, essentially what you end up with any of these methods is you get something akin to like an n-dimensional image. And so instead of, in contrast, like a regular, image you might be used to, which has a red, green, and blue channel, each of these images, regardless of the data type, you essentially end up with a stack of n channels, where each channel in this stack corresponds to like some biomolecular species you're looking at. And so the first thing we do is we take these different data sets and we condense them down into sort of categorical features at 3 spatial scales. And so the first spatial scale is all the way down to individual pixels. And so essentially by looking at how different, in this case, different proteins overlap and which ones co-occur at a pixel-based level, we can then categorize different pixels into pixel types. And so for instance, in this example, we see that there is a predominant pixel type that's shown in brown, where 3 immune markers align there. And so we can then go from the pixel-based categorical features to things that are cell-based. And so after we've run these through a few deep learning pipelines that identify the borders of cells, we can look at where those borders are relative to the different pixel types. And we can start to make sort of meaningful assignments about sort of biologically informative cell types. And so for instance, those 3 immune markers that we were showing there, the way they were co-expressed sort of corresponded to macrophages. And so you can see there's the yellow cells here. And then once we've got sort of these features first at the pixel level and then at a cell level, sort of like the next layer of like the nested Russian doll is to think about sort of how all these things are sort of spatially laid out relative to one another. And so essentially at that point, we start to look at sort of how different cell types are spaced out. And so in this case, we're doing something called a neighborhood assignment, where essentially we're looking for groups of cells that tend to occur close in space. And in this case, in this inset, you can see that little area that I was showing before was enriched in sort of neutrophils and macrophages. And through this approach, that was one of the neighborhoods that came out. So we have sort of this sort of nested hierarchical way then of sort of building up this representation of the images. And ultimately, all of that goes into sort of extracting quantitative features that build out these spatial profiles. And so once we have those spatial profiles, we can interrogate them with different quantitative pipelines to really understand what's going on. And so as the first example of how we do that, this is one study we've been looking at, studying glioblastoma multiforme, which is a grade 4 CNS lesion. The survival is really, really kind of awful, and despite a lot of targeted therapies, it really hasn't gotten much better. And so there's a lot that's in this study right now, but I wanted to kind of focus on one example of some of the stuff that we found with respect to antigen expression of different proteins in these tumor cells that are currently being targeted with specific sort of antibody therapies. And so as part of that spatial proteomic imaging that we did for this study, that included 7 proteins that are drug targets. The most abundant are B7H3 and EGFR. And so I'm kind of showing here just 6 examples of different patient samples, and you get of tumors from different patients. And you can see that like even with just looking at these 2 proteins, there's pretty wide diversity of how they're co-expressed, what their spatial distributions are. And that continues on if you look at like all 7 of these targets. And so there's a lot of different permutative states that you see at a single cell level. And so this is just showing for each one of those 5 patients, for 100 cells from each patient, which of these protein targets are present or absent. And so we can see there's a lot of cells that express more than one protein. And we can now aggregate that into, a histogram to sort of say, okay, how many tumor cells express 1 antigen target, 2 antigen targets, so on and so forth. And the first thing that we see is that around 63% of tumor cells express 2 or more antigens. So once we have that information, we can start to kind of have some thought exercises about how this might impact targeted therapies and how we might improve them. So tumor coverage here, I'm just using to refer to the percentage of tumor cells in a patient sample where, per our analysis at least, what percentage of the tumor cells express an antigen that's the subject of a targeted therapy. And so, so then if we want to have a goal that we want to administer a drug regimen that's going to target 90% of tumor cells, then we can ask the question of sort of, if we're using single agent therapy, how many patients would meet that threshold? And we find that it's not that many. It's probably around, right at 30%. And so then the question is like, okay, well, if we then say, okay, we're going to administer a 2 agent therapy where the 2 drugs that we're selecting are the 2 most abundant for that patient, does it do we improve? Do we have more patients where we get hit 90% tumor coverage? And the answer is yes. So it's like around 21% at that point. So that's good. And so, we saw this as like, oh, this is great. Let's just keep increasing drugs. We're going to, hit all the tumor cells. But so that's kind of the question we thought, because you see here the trait that we see with 2-agent therapy is the reason why you start getting a higher percentage of tumor cells is that even though a lot of the tumor cells co-express these targets, there's the majority of tumor cells only express one or the other. And so the example that we're given here is with patients that were the 2 most abundant antigens were B7H3 EGFR. So then, okay, so what happens if we increase to a larger number of targets? Do we get more patients over this 90% threshold? And unfortunately, it looks like not really so much. So even if you're to have a 7-agent therapy, you would still only have that would still only hit 90% of tumor cells in around 60% of patients. And so 40% of patients, even with that many, would fall below that sort of arbitrary threshold that we had for this thought exercise. And the reason is that the vast majority of patients have tumor cells that are sort of in GBM have tumor cells that are pan negative for all of the agents that we're looking at right now. And this kind of goes into a broader issue with gliomas, which is that there's a very immune suppressive TME. This has been known for a long time. And, and what this says to us is that any of these drug therapies, targeted drug therapies, are probably not going to be very efficacious unless we can, at the same time, sort of induce the immune system to, be able to clear out tumor cells that are negative for all these targeted therapies. And so along those lines, one of the things that's been implicated as a big driver of immune suppression and the glioma TME is aberrant glycosylation. And so we took these same samples and we analyzed them with MALDI to be able to look at sort of, with respect to how severe the tumor was, do we see sort of differential enrichment at a patient level on specific glycans? And so we were able to take around 118 glycans in this analysis. And we found, like, remarkably, just looking at the glycans, not even taking into account the spatial distribution of them, we get actually pretty accurately assigned glioma grade purely based on the glycans. And so this doesn't even incorporate any protein information or anything. And the glycans that seem to be the most discriminative for these are a class of glycans called sialoglycans. And so they have, and these are called that because they have sialic acids on them. And these glycans are of a lot of interest right now because they are ligands for a family of receptors called Siglecs, which can have functions when they're present on immune cells that are very similar to what you have with, like, sort of immune checkpoint inhibition. And so we're kind of looking into this more in the context of a lot of different treatment trials. And so we're going to look sort of how these different antigens evolve and change with recurrence and immunotherapy. We're also going to be integrating all of these things together so we can actually have a true multivariate view of what's going on with grade and with disease progression. And then we're going to be making all of this available online as soon as we get a preprint up, hopefully in 2 or 3 weeks. So anyone that wants to be able to look to see how these different factors vary with respect to different patient covariates, whether it's, age, tumor grade, time of recurrence, or anything like that, can go on there and interrogate all this for themselves. So that's kind of the first cancer example. The second one I'm going to tell you about is one in breast cancer. And this was a part of the Human Tumor Atlas where we looked at ductal carcinoma in situ. So DCIS is a pre-invasive form of breast cancer where the tumor cells are sort of separated by the surrounding stroma, which we've highlighted here in brown, by this concentric layer of myoepithelium. So it's kind of like, you can think about it kind of like a ball with a layer of myoepithelium around it. And this is really the distinctive thing that makes it not invasive breast cancer. So it's a pre-invasive cancer. So those cells are tumor cells, but they haven't yet invaded the stroma. And so this affects about 60,000 women in the US each year. It's life-threatening only if it transitions to invasive breast cancer. And the issue is that probably about half of women, and this is, a lot of this is based sort of on retrospective studies and but in Europe and in the US, about half, if we didn't treat them, would probably never progress to invasive breast cancer. But because at the time we can't prospectively identify these women at the time of diagnosis, vast majority of patients are still treated sort of presumptively as if they will be progressors. And so, as you can imagine, this means that we're very likely giving a lot of unnecessarily aggressive therapy because we're not able to stratify these women at the time of diagnosis. So the big question we had was whether or not we could go and take a bunch of tissues from archives at Washington University, where we have the outcome of how these women did, whether or not they progressed to invasive breast cancer. And can we go back to those original primary tissue specimens that were acquired at the time of a DCIS diagnosis? And can we use this approach to be able to risk stratify based on that endpoint data? And so we went through and we created these spatial profiles. And essentially, there's kind of three major insights that we pulled out of this study. And so the first is that if we went and we asked the question of which of these features within that spatial profile seem to segregate with outcome, the first thing that we found was that the spatial organization actually ended up being a very, very big factor into which variables were the most important. So, and so what I mean by that is that, so you can imagine, for instance, if we have an image of a tumor, we could have parameters that we pulled out of there that really didn't require spatial information. So for instance, what percentage of all the cells were tumor cells, right? That's just kind of a number. It doesn't really take into account where the tumor cells were or anything like that. But then we can also have features that are informed by the spatial construction of the sample. So for instance, what is the average distance between a macrophage and a tumor cell would be one. And what we found is that even though the split between those is about 50-50, if we looked at the top 20 most important features, the vast majority of them had some sort of spatial aspect to them. Sort of speaking to that it's not enough to know just like what something is, you really need to know where it is relative to other things to be able to get these, more informative clinical prognostic predictors. And so the second thing that we found was that there, also sort of to support this idea of we should really be looking at tumor lesions as more of like an ecosystem, is that if we also look in those top 20 features, there really wasn't any one single cell type that was, broke the case wide open, right? It was actually a sort of a cumulative effect of around 14 distinct cell populations and what the spatial distributions of these were. And the one that seemed to be the most important in there was myopithelium, which was this layer that is talked about that's surrounding the tumor cells. And we found that changes in that myopithelial layer were actually tightly correlated with, if you looked in the surrounding stroma, of sort of how reactive that stroma was, how many immune cells were in there, and also other things like whether or not there was a lot of hypoxic signaling going on. So to follow up on this, we've recently expanded this effort to include a lot more patients. And we've kind of we have a sort of 2.0 version of this spatial study. And using that data, we've now found that we can do a global risk classifier on these retrospective samples where we can achieve an AUC of around 0.82. And that's predicting whether or not women will recur within 10 years. And so, so it's pretty remarkable. The AUC for this at this time, I don't think is, it's probably not at the level that you would need for it to be to actually be, I think, clinically impactful quite yet. But I think what it does show that we're getting more confident about is that there is information within the spatial distribution of these samples that seems to have very long-term prognostic significance, which is very interesting. The other thing that we found that I think might be where we're trying to move in the near future in terms of trying to find something that can be translated clinically is this idea that a global risk classifier is kind of one thing, but the other thing is that can we identify a set of features to where we can just say for maybe 20 to 30% of women with very, very good negative predictive value that you are not going to recur. And we've actually found a subset of features that we think might be very good for that. So, if we're not able to get something that's sort of useful and pragmatic in terms of global stratification, we think we might at least be able to come up with a score to assign a low risk population. And so we're diving into that a little more now. So with that, now I'll jump. So the thing is with cancer, this is sort of, if you look at this, all these things are sort of emblematic of cancer, immune suppression, Warburg effect, ECM remodeling, endogenous retroviruses. And remarkably, all of these are present in pregnancy as well. And the parallels are pretty remarkable. It's been known for a long time that the degree of placental invasiveness in mammals correlates well with which cancers that mammal is susceptible to. And humans have one of, if not the most invasive placenta of all mammals. And what goes along with that is sort of we are susceptible to certain types of cancers that other animals don't get. So for instance, cows don't get melanoma, we do. And there's actually some really great studies out of the Gunther lab at Yale, where they've been able to show that that is most likely because of evolutionary features where the human endometrium has evolved to be permissive to invasion by the placenta, and that this can explain why melanoma cells are actually able to invade fibroblasts in humans better than they are in cows. So, placental trophoblasts are in many ways a tumor cell surrogate. They're invasive, they're genetically dissimilar, and if you look at sort of the way the placenta attaches to the endometrium, it very much resembles in many ways from a histological standpoint an invasive carcinoma. And there's even this nice sort of well-organized spatial trajectory of differentiation where you start with villus trophoblasts and you start to see the emergence of extravillus trophoblasts invading the endometrium. And so these EVTs, they bud off from the villi and they invade into the decidua, and they have really remarkable and nuanced interactions with the maternal immune cells and the stromal cells, and also they coalesce around arteries within the decidua and participate in this crazy process where, along with contributions from maybe decidual NK cells, the spiral arteries within the decidua lose their smooth muscle, and they become much, much larger than they were in the non-pregnant uterus. And to make it even stranger, the EVTs actually invade the arteries and replace some of the endothelium within the arteries. And so the fact that this is done and this has happened and that this occurs and is immunologically regulated to occur, we thought is incredibly fascinating. And so so that's kind of, with our study, this is kind of what we really wanted to understand, was sort of this process and how this evolves in the first 20 weeks. And this has a lot of bearing, also understanding this process, besides being biologically terribly interesting, it's been implicated in a lot of different pregnancy disorders as being dysfunctional. And I think the canonical one is preeclampsia, where it's been noted for a very long time that spiral artery remodeling seems to be subpar and does not advance to the stages that it does with you see in decidua from term pregnancies of normal pregnancies. So with that in mind, and what we really wanted to do was understand and sort of comprehensively define the structure and composition of decidua in humans by leveraging like tissue that's just present and archives within different pathology departments. And then we really wanted to try to extricate and tease out two different things, because this process is kind of interesting because there's clearly local effects that invading trophoblasts have on how this proceeds, particularly with spiral artery remodeling. But there's also then this temporal component that, there's also sort of a time-dependent effect of pregnancy. And so you have like two kind of axes of regulation, right? You have something that's kind of spatial, but you also have something that's temporal. And so we wanted to find a way to be able to kind of, as best we could, extricate these things in this study to understand what could be attributed to what. And so this was a study that was led by Shirley, Aaron, and Inna. And essentially what we did was we took these samples from around 66 different patients. We isolated regions within the decidua that contain spiral arteries, and we subjected them serial sections to two different analyses. The first was using spatial proteomics with MIBI, and the other was using spatial transcriptomics. And so kind of to start off with the first, once we have these multiplexed images, and here's kind of showing an example of one with decidua, we subject them to cell segmentation, and then we can cluster all of these cells into sort of major cell types. And in this study, the majority of the different cell populations were maternal immune cells, and then the second most, and then the other major group that we're very interested in was looking at these different invading trophoblast populations. So once we have those, we can map them back onto the tissue and see exactly where all of them are. And by sort of looking at their proximity to arteries, how those arteries are changing, and then taking into account what gestational age the sample is, we can start to kind of, slowly untangle this ball of yarn to see what seems to be driving what. So the first thing that we want to do specifically for this study was to be able to sort of quantitatively characterize these sort of well-vetted changes in sort of artery morphology. And so this is what an artery, an unremodeled artery looks like. So you can see that they're small. They have a concentric layer of smooth muscle going around them, which you can kind of see here in that salmon color. And then on the inside, they have sort of intact endothelium lining that vessel. And so by stage 2, you can see that that the smooth muscle has started to sort of fragment and balloon up. That's sort of signs that it's undergoing apoptosis and the vessel is starting to get bigger. By stage 3, most of the smooth muscle is gone, and you can see that the endothelium is kind of taking on a little bit of a cuboidal shape, and these purple EVTs are now starting to sort of coalesce near the artery. By stage 4, the EVTs have actually entered the artery, and they're forming these large clumps and balls. And then by stage 5, you have a fully remodeled artery where all the smooth muscle is gone, the vessel's much, much larger, and you can actually see that there is the trophoblasts have replaced some of the maternal endothelium here. And so so the pictures are really nice, but what we wanted to be able to do is sort of extract quantitative features from these so that we could score them in a more rigorous way at what stage they were at. And so to do that, we essentially we quantified the different morphological features here, like for instance, how continuous the endothelium was within the vessel, how thick or fragmented the smooth muscle was, whether or not there were EVTs in there, and so on and so forth. And we extracted all those and essentially built a model so that we could give it a simple score from 0 to 4 or 5. That would a continuous score that would tell us like how far along these arteries were in the remodeling process. And so once we did that, the first thing we asked is it's like, okay, how does the stage of artery remodeling sort of trend with gestational age? And you can see here that there's definitely a correlation, whereas you get to later weeks, you see more and more fully remodeled arteries, but it's also not kind of a one-to-one. There are patients in here that even at weeks 8 or 10 had stage 4 or 5 remodeled arteries. And so they're correlated, but there seems to be, as people have said before, sort of this degree of local regulation. And we can see that here because now that we have the gestational age and we have this quantitative score, delta, we just, with simple linear regression based on the data that we got from the spatial transcriptomic data, we can ask by comparing the changes in those genes. We can ask, how do those genes, what do they seem to be more correlated with? Do they seem to be more correlated with the stage of the spiral artery, or do they seem to be more correlated with the gestational age? And you can see that, the vast majority of genes in the artery seem to be, much more correlated with what's going on spatially around them. And with that, we were able to actually pull out specific gene modules that sort of typify each of one of these stages in spiral artery remodeling. On the flip side, there were definitely aspects of the decidua that were not spatially regulated, and one of those was the sort of immune composition. And so we found that based on just the maternal immune cells, the frequency of those immune cells, we could predict what the gestational age of that sample was within about 10 or 15 days. And so now I'm just going to share you sort of one of the things we asked the question of was, can we say something based on these data about what the potential path of invasion of EVTs are? And so there's been 2 routes of invasion that have been proposed. The first is that they invade into the decidua and they migrate to the outside of the artery, and then they kind of punch in to the inside of the artery. The other is that they actually migrate up the artery at the intravillous space. And so we wanted to see if based on sort of the temporal ordering and the spatial layout of different cell populations, could we get an answer into which one might be going on? And so what we did was we looked specifically at trophoblasts right at the cell column when they're first invading the endometrium. We looked at them, we looked at their phenotype when they were within the decidua versus when they were right around the outside of the spiral artery. And then we also looked at them when they form these clumps within the artery. And so the first thing that we found was that if you look at the sort of change in phenotype with those, you see a trend. You see like sort of a fairly continuous trend in how they express different proteins that go along with this path of going into the decidua, going around the artery, and then into the vessel. When we did spatial transcriptomics, we really wanted to understand exactly what was going on in this transition from the intravascular, from where they're outside the artery to when they go in. And the cool thing is that we found there's a dramatic change in sort of what proteins they're expressing. When they're inside the decidua, they're mostly expressing things that have to do with sort of tolerizing antigen presenting cells, motility proteins and factors. Whereas when they go into the arteries, most of those kind of go down. And what you see is a lot more angiogenic factors, and then things that are really known to prevent macrophage phagocytosis, as well as downregulation of the maternal immune system. So again, it almost mirrors a lot of what we think happens with tumor cells when they undergo angiolymphatic invasion. And so the other thing that we can ask, right, was that if the EVTs are coming in and they're starting the outside of the vessel and then they're going in, one of the things that you would probably expect would be that you would need to lose that smooth muscle layer around there before you would start to see EVTs inside the vessel. And that's indeed what we found, was that, if you look at sort of the number of EVTs within vessels, that essentially it was restricted to arteries that had almost lost all of their smooth muscle. So we didn't see any where there were trophoblasts inside the vessel that had a lot of smooth muscle. And we also saw a similar trend sort of with endothelial loss, which is you don't really start to see EVTs inside of arteries until you see gaps within the endothelium. And so this, once again, doesn't prove, but sort of circumstantial evidence to suggest that, the loss of that concentric muscle layer and breaks within the endothelium are highly correlated with whether or not you see EVTs within these vessels. So, so it kind of supports this process of it being through the decidua, through an intravasation mechanism. In the last few minutes, I was just going to show you briefly some stuff that we've done looking at glycosylation in a similar manner that we did with these gliomas. And so because we've got, these stepwise changes in terms of EVT invasion, we thought by looking at those EVTs in the context of what glycans they express, it could maybe give us some insight into how these glycans are driving EVT invasion and maybe even immune invasion. And so glycans are a highly diverse set of PTMs. I kind of won't belabor this. This slide's more to say it's like, wow, there's a lot of different shapes, and there's a crazy number of glycan structures. I don't even know if we fully know the full scope of this. But we do know is that there are fairly well conserved functions that segregate with different glycan classes. And so what we did here is we took these same samples. We did a stain to be able to see where the trophoblasts are. And then on that same slide, we did multi-glycan imaging. And essentially, then we co-registered the two so that we could see which glycans spatially co-registered with where the trophoblasts were in that core. And the cool thing is that we found that just similar to the glioma, based purely on the glycan composition for where these EVTs were, these different regions, we could actually classify whether or not that region was attaching EVTs, EVTs that were invading the decidua, or EVTs that were even within blood vessels. And once we went through and we clustered these together, we saw that highly branched glycans seemed to be a big driver of sort of how these different, how they were separated along this differentiation trajectory. And so by then going and looking back at the spatial transcriptomics data we did, we saw what the glycans were that seemed to be driving the separation. There's a lot, we already know from the literature which enzyme should be involved in the synthesis of those glycans. And so we went to see if that kind of lined up with what we were seeing here by looking for transcripts for the glycosyl transferases that are involved in those enzyme pathways. And sure enough, we found this one called MGAT5, which is involved in making this sort of fourth branch on these tetraantennary glycans. And the interesting thing is that we found that this MGAT5 has actually been shown in tumor models to be a big driver of invasion through the production of these glycans. And so it looks here like, you know, this, we're starting to wonder if this might actually be sort of the evolutionary root of why we're making, you know, why we even have the capacity to make these glycans that allow us to invade locally and you know, evade the immune system, which is that it could very well be a critical part of being able to facilitate placentation and placental invasion. So with that, I can kind of finish up here. You know, this is hopefully showed you like how we can use different spatial techniques to make inferences about function and primary human tissue. You know, the big part of this with any of these multiplex techniques is they just need to go along with sort of automated analytical pipelines so that you can look at things at scale. And sort of thoughtful integration of different modalities, I think will kind of, I think is very promising for us being able to figure out things that we couldn't really know just by looking at a single method. And so with that, if there's any time, I'm happy to take questions.
Great. Thank you so much, Mike. And we do have about 5 minutes. If anybody wants to just go ahead and unmute themselves, happy to have you ask a question. I don't see anything.
Hi, should I just start talking? I raised
Yes.
my hand, but then you said to just start talking. Hi, Michael. Thank you. That is really amazing work. And what I was wondering was you talked about intravascular trophoblasts, and it was my understanding that there are these really flat trophoblasts that kind of become the new endothelial cells, but there's also these really puffy, big trophoblasts that are inside the vessel. And so I was wondering if your analysis, when you call them intravascular, are they those puffy ones or the flat ones? Or if you distinguish, if those are distinguishable transcriptionally?
No, that's like a great question. And so, and I think, so the first thing that you see, the puffy guys are, I think, the first ones you see, because that's kind of the morphology of these large clumps of EVTs that you see in the arteries sort of earlier in spiral artery modeling. Those clumps sort of resolve and go away around like week 16 to 20. And that aligns with when you see the appearance of these more flat EVT phenotype that is lining at least partially the vessel. That's the endothelialized EVT phenotype. We collectively were referring to anything within the vessel as intravascular, so either both types. Something that we did not do that I wish we would have done is would have been to actually, because all of the transcriptional stuff that we've seen that I showed is actually the clump larger cells. We did not collect transcriptional profiles because of like technical issues with trying to isolate them with nanostring DSP, the endothelialized ones, which are really kind of thin and narrow. But it's something the techniques around this stuff have improved a lot, and it's something we actually want to go back and do.
Great, thank you.
Great. And then, Mike, any insights into from the data of the type of immune cells? It sounded like the proportion of immunes is very gestationally age driven. But any spatial relationship between the type of immune cells interacting with each other versus interacting with the EVTs?
So there was one population of NK cells that we found had a very striking accumulation around the arteries when they were at stage 2, and then they subside. And these were ones, I'm trying to remember. I have to go back and look at the paper. I think they were NK cells that were CD57 positive. And we actually got a lot of grief from the reviewer, because they said that meant that they were derived from peripheral blood. And so we had to go back and do like all these confirmatory experiments to demonstrate they really were CD57 positive and all these kinds of things. And then we also saw there's a lot of preferential spatial neighborhoods that we saw with there was a macrophage subset in there that seemed to be interact pretty frequently with EVTs. So I think, part of this is that I think if we were to go back and do this again, you know, hindsight's 2020. The regions that we looked at were sort of tightly focused around the spiral arteries. I think the smarter thing to do would have been to do in retrospect is for us to have also collected decidua that was very distant from arteries as a comparator, because one of the things I suspect is that some of these interactions may be fairly, you know, they may be pretty enriched within a, you know, 0.5 millimeter of an artery, but you would see a depletion of them if you looked at sort of decidua that was far away from the artery or far away from the, you know, point of attachment. And that really wasn't included here. So we didn't have that contrast to really see some of these relationships. And so it's something that, you know, I'm hoping we can go back and look at some point. Great.
Well, we are at time. Thank you, Mike, for taking us into other worlds other than the placenta.
Virginia, Roger had one quick question.
Oh, excellent.
Yeah, just a quick question, Michael. Very lovely presentation. Just wondered if you had a data set on preeclampsia that you could compare with this lovely outcomes.
We do, and we're teasing through it to try to understand what it means. There's some very interesting changes, and that is entirely because of Virginia held our hand to put together this tissue microarray that has preterm preeclampsia with some preterm, you know, lack of a better term, controls. So preterm without preeclampsia, as well as term preeclampsia and you know, sort of normal term birth. We're really seeing some pretty striking differences is in the comparison of those preterm preeclampsias that pretty much every other group. And there seems to be, and we're still going back and verifying all this right now, but there seems to be a non-insignificant drop in some of the glycans that we identified as potentially promoting invasion in the anchoring villi. Now, you know, the thing is that it's, I think, you know, I mean, preeclampsia is so tough. It seems so impenetrable in many ways. And, you know, and we're trying to think of ways that we could sort of get a better idea if this is sort of correlative and a consequence of what's going on, or if it's actually, you know, tip of the spear for why you sometimes get this more like shallow invasion with preeclampsia. And the issue, and the thing is this is where like that ball of yarn comes back into play, because so much of like glycan synthesis, right, is coupled to sort of oxygen status and metabolism, right? So it's sort of, which is also known to be perturbed. So it's just, yeah. So so we're kind of moving slower on this to try to think about what would be to really thoughtfully figure out what these data are telling us and what the next best step is. But we are looking at it hard.
Sounds like you and I need to get together and talk more.
Yeah, absolutely. We need to do that.
All right, very good. And I'm fascinated by the glycan too, because there's some glycans that we've identified are very highly regulated in preeclampsia. So I'm.
Oh, really? Oh, well, maybe this isn't nothing. Maybe we should definitely get together. And
I think we need a joint lab meeting soon.
No, we do. Yeah, it's because it's just sort of, you know, because we're you know, preeclampsia. It's just, gosh, it's sort of by nature of everything about it. It's just, you know, there's not any good animal models. It's just, I mean, it's just, it's very hard to get at. So I think we got to bring everything to bear on it, I think, if we're going to figure it out.
Yep. Great. Well, thank you all for attending. And thank you, Mike, again, for a great talk. And look forward to everybody next month.